Let's talk about some weird viruses, because there are a lot of them. First up, Bacteriophage Lambda. So Bacteriophage Lambda, small dick energy. Anyway, Bacteriophage Lambda infects bacteria, specifically E. coli, and it can either do the lytic or the lysogenic pathway, but the way that is chosen is so overly complicated. We have a lot of proteins involved. First up is C3. C3 is regulated by the host cell health. If the host is healthy, there is a lot of C3, which is good because we need C3. What C3 does is it protects C2 protein from the host cell protease, which is a specific enzyme that just degrades protein, and the virus does not want its C2 to be degraded. So C3 protects C2. What C2 does is it helps regulate C1 protein, and what C1 protein does is it suppresses a bunch of the other viral proteins. Those viral proteins are what sends it into the lytic bursting pathway when those proteins are active. So C1 stops the cell from bursting. There is another protein, CRO. CRO represses C2, which represses C1. If there is too much CRO, C2 gets repressed, which makes C1 stop working, which makes the cell burst. But CRO is regulated by C1, so if there is enough C1, there is less CRO. So CRO does not suppress C2, which just helps with C1. So C2 regulates C1, C1 regulates CRO, CRO suppresses C2. Now, when the host bacteria gets stressed or damaged or otherwise bothered, the bacteriophage does not like that very much. When the cell gets stressed, C3 production goes down. Since C3 is down, C2 gets degraded by the host proteases, and so therefore cannot regulate C1. C1 is down, which produces more CRO. CRO, in turn, regulates whatever C2 is not degraded by the proteins, which stops the regulation of C1 even more, so even more of the lytic proteins are made. And so eventually gets to a point where the cell is like, nah-uh, and bursts. Complicated? Yes. Overly so? Yes. Welcome to virology.